I was just reading the latest clinical data on protein quality control in the brain, and it changes how we should evaluate cellular clearance decline. The brain must constantly clean itself to function. We often assume that age-related memory issues are caused by structural loss. However, the cellular reality is that a slowdown in removing waste might happen before any permanent damage occurs. Here is the mechanistic question. What happens when cellular clearance systems fail? By examining this specific protein quality control network, we can find out how cells protect their vital synaptic connections. Neurons require highly active transport systems to maintain protein quality over long distances. Unlike shorter cells, a neuron must constantly move waste across its extensive axonal networks for disposal. When this complex network of protein maintenance slows down, cells accumulate damaged proteins. This gradual accumulation can create localized cellular stress, disrupting normal communications before these cells show any structural decline. We need to understand how the brain's internal cleaning crew operates. This allows us to see how maintaining clear pathways is essential for preserving cognitive performance as we grow older in age. In the aging nervous system, protein quality control systems gradually decline. This cellular slowdown causes damaged proteins to build up, leading directly to aggregates that disrupt normal chemical signaling. This accumulation of misfolded proteins is not just a side effect of aging. It represents a progressive bottleneck where cellular clearance mechanisms can no longer keep up with waste production. When waste builds up, it impairs synaptic transmission. Follow the evidence boundary. This cellular slowdown is what we eventually observe as a gradual decline in memory and processing speed. Now, the important mechanism to analyze is proteostasis, which is the cell's system for balancing protein creation and removal. Aging directly degrades this delicate, complex protein quality control network over time. As proteostasis declines, the cell's ability to fold new proteins correctly is compromised. This cellular dysfunction creates a high concentration of unstable, sticky proteins that are prone to sticking together. These sticky, misfolded proteins aggregate into larger structures that the cell cannot easily break down. This aggregation blocks essential transport channels, leading directly to a severe, progressive cellular logistics crisis. To understand this crisis, we must define the proteostasis network. This complex system contains specialized chaperone proteins that help fold new molecules and cellular clearance systems that destroy damaged ones. Chaperones act as the first line of defense, preventing proteins from folding incorrectly. When chaperones are overwhelmed, cells must rely entirely on their clearance systems to remove all the debris. The primary clearance systems are the ubiquitin proteasome system and autophagy. The proteasome degrades individual small proteins, while autophagy clears much larger aggregates and also whole damaged cellular components within the cell. In a young brain, these systems maintain a clean environment. However, as cells age, both proteasome activity and autophagy efficiency decline, allowing these damaged proteins to persist much longer in the cytoplasm. This persistence changes the cellular environment, making it more favorable for protein aggregation. When individual misfolded proteins remain in the cell, they bump into each other and form toxic clusters. These small clusters, called oligomers, are highly reactive and can cause cellular stress. Over time, these oligomers grow into much larger, insoluble fibrils that are very difficult for the cell to clear. The transition from soluble proteins to insoluble fibrils represents a major change in cellular health. It marks the point where the cell's self-cleaning capacity is officially overwhelmed by this accumulation. What matters clinically is, this buildup is a slow process that occurs over decades. By studying these early stages of proteostasis decline, we can understand the tipping point of brain aging. Now let us examine the proteostasis cliff, which is the tipping point of cellular clearance failure. Initially, cells manage to compensate for this gradual decline in their protein quality control. Chaperones work harder, and the cell increases its autophagy activity to handle the rising waste. This compensation phase keeps the cell functioning normally, hiding the underlying decline in clearance capacity. However, this compensation has a limit. When the accumulation of misfolded proteins exceeds a critical threshold, these clearance systems become saturated, and waste removal drops off very rapidly in the cell. This sudden drop in clearance capacity is the proteostasis cliff. Once a cell crosses this threshold, even a small increase in protein damage leads to a massive accumulation of aggregates. The accumulated aggregates then begin to actively inhibit the remaining clearance systems. For instance, large protein aggregates can physically block the proteasome, stopping it from degrading other damaged proteins. This inhibition creates a dangerous feedback loop. More aggregates lead to slower clearance, which in turn causes even more aggregates to accumulate, accelerating the decline of the entire cellular environment. As this feedback loop continues, the cell's internal transport system is disrupted. Proteins and organelles can no longer move efficiently between the cell body and these distant active synapses. Synapses, which require constant protein renewal to transmit signals, are the first to suffer. Separate the mechanism from the outcome. This local communication pathway slowly fails over time. This synaptic failure is highly localized at first. Individual connections go silent as their local protein pools are depleted and local clearance systems are overwhelmed by these sticky, misfolded aggregates. Over time, these silent synapses accumulate, leading to a measurable decline in cognitive networks. The brain loses its flexibility, and forming new memories becomes much more difficult for the organism. Furthermore, the accumulation of aggregates triggers a cellular stress response. This response is meant to protect the cell, but when activated long-term, it can halt all new protein creation entirely. Halting protein synthesis prevents the cell from making new aggregates, but it also stops the production of essential proteins needed for synaptic function, causing a further localized communication breakdown within the brain. The cell is trapped in a state of chronic stress, unable to clear the old proteins or make new ones. This state is consistent with cellular senescence, where cells survive but cannot function. This mechanistic progression explains why cognitive decline can accelerate so quickly once it starts. The crossing of the proteostasis cliff triggers multiple self-reinforcing pathways that drive rapid, progressive brain decline. By understanding this cliff, we see that aging is not a uniform slide. It is characterized by threshold events where small cellular changes lead to large shifts in cognitive function. The important limitation is, not all cells in the brain experience this decline at the same rate. Some neuronal populations seem to maintain their clearance capacity much longer than others. A plausible counterpoint is that protein aggregation might sometimes serve a protective role. In this view, clustering misfolded proteins could be a way to sequester toxic molecules from the cell. This means that simply dissolving aggregates might not always be beneficial. If the cell's underlying clearance pathways are still broken, releasing these sequestered proteins could increase local active cellular toxicity. Consider a validating case where a cell maintains proteostasis under stress. In this hypothetical model, increased chaperone activity helps prevent protein aggregation, demonstrating that protein folding stability is crucial. Furthermore, this clearance process is highly dependent on overall cellular energy. If energy production is compromised, even a healthy proteostasis network may fail to keep up with essential protein turnover. This suggests that protein quality control is not an isolated system. It is deeply connected to the cell's metabolic state, which must be considered when evaluating our own cognitive aging. Therefore, we cannot look at protein aggregation in a vacuum. We must evaluate it as part of a wider network of cellular health and energy production within our brain. We can now resolve the question of why damaged proteins build up. It is the result of a progressive imbalance between the rate of protein damage and cellular clearance capacity. As we age, daily cellular stress causes proteins to misfold. At the same time, the cellular machinery responsible for clearing these damaged molecules becomes less efficient and slowly degrades over time. This dual change creates a slow, steady accumulation of waste. Initially, the cell compensates, but eventually it reaches the proteostasis cliff, where the cellular clearance network is completely overwhelmed. So the buildup is not a sudden event, but the crossing of a critical threshold. Once reached, the accumulation of misfolded proteins accelerates rapidly, leading directly to macroscopic cognitive decline. This mechanistic understanding helps explain why cognitive aging can progress in steps. Each step may represent a local tipping point where a group of active cells crosses their clearance threshold. By focusing on this tipping point, we can shift our perspective. Protect your brain. We must view decline not as inevitable decay, but as a series of manageable local cellular thresholds. This shift allows us to design better ways to support the brain. We focus on maintaining the efficiency of the proteostasis network before it reaches its critical high saturation point. This approach targets the root cause of the buildup rather than the symptoms. By supporting the cell's natural cleaning pathways, we may help preserve this synaptic communication much longer. The key is to intervene early in the process. We must support the proteostasis network while the cell is still in its compensation phase, protecting the entire system from overload. To apply these concepts, we can focus on general low-risk lifestyle habits that support cellular health. For example, regular physical activity may encourage natural clearance processes in our cells. Another important behavior is prioritizing quality sleep, which is when the brain's waste clearance pathways are highly active. Quality sleep can help the brain clear daily protein accumulation. Additionally, maintaining stable metabolic health is crucial. Because clearance systems require significant energy, supporting overall cellular energy production can help maintain these complex, vital protein quality control networks. We can also observe our daily routines and identify areas to reduce chronic stress. Reducing stress may help lower the rate at which these proteins become damaged and misfolded over time. This approach is not about finding a single unproven solution. It is about implementing low-risk lifestyle habits that support the human brain's natural maintenance systems over time. What matters clinically is, these adaptations do not constitute medical advice or treatment. They are educational concepts meant to help you evaluate your own daily health habits and patterns. Consult a qualified healthcare professional before making any significant changes to your daily routine. This ensures that any adjustments you make are safe and appropriate for your body. By focusing on these fundamental cellular processes, we can take a proactive approach to brain health. We support the systems that keep our neural networks clear and functioning over time. We must evaluate crucial cellular clearance decline by looking at the active local proteostasis networks. Optimize the protocol, protect your brain, and I'll see you in the next data set.